everyone, my name is Fikri, a third-year PhD student. The work I will be presenting today was done at the BRGM and ISTO and was co-financed by BRGM and ADEM. France is a country that is suffering from urbanization as soil artificialization have reached 9.3%, which means that France loses the equivalent of one football stadium of natural or agricultural soil to artificializations every 5 minutes, and the equivalent of one county every 7 years. One of the solutions to tackle this problematic is to reuse abandoned brownfields that are usually characterized with degraded soil. In order to do so, we have to first rehabilitate the soil and ensure its durable refunctionalization. Soil is a living system where many interactions occur between the biotic and abiotic components of the environment. The presence of the right physicochemical parameters, such as nitrogen, carbon, and phosphate contents, yields to the development of organic forms of life, like microbes or meso and macrofauna. These organisms occupy ecological niches and accomplish some activities or functions like storage, recycling of organic matter, storage circulations of water, and storage, supply, and cycling of nutrients, which will be our main focus, especially the nitrogen cycle, due to its importance for ecosystem functioning, as it helps plants synthesize chlorophyll, enriches the soil by the conversion of inert nitrogen gas into usable forms by plants, which helps them grow. Here we can see the site that was chosen for our study, which is Pierre Benit site located in Lyon, France. This site was rehabilitated in the frame of BioTubes project in 2016 using different restoration methods. 4 plots were sampled for the present study, which are AS for initial soil, which serve as control, which is coded in blue, LO1 that is coded in green, LO2 coded in yellow, are both composed from the same technosoil with low organic matter, but have received 2 different types of plantations and bacterial inocula during their rehabilitation. Finally, we have HO, which is coded in orange. It is made from a different technosoil, which is rich in organic matter, but costs more than the others. So the idea is to test if the most costly method, which is HO, is more resilient and more resistant to perturbation post-rehabilitation. The main question investigated in my study was what is the most optimal rehabilitation process for derelict urban soils. To answer this question, we conducted a laboratory experiment which aimed to test the resistance and resilience of soil functions post-heat and drought stress in urban soils that were submitted to different rehabilitation trajectories. We characterized the general microbial activity along the experiment by measuring basal respiration and extracellular microbial activities like phosphatase and beta-glucosides. Finally, we focused on the end cycling functions, specifically nitrification and denitrification. Microorganisms are the primary contributors to nitrogen cycling in soils. They are directly responsible for nitrogen fixation from air to soil in the ammonia form. Ammonia is then transformed to nitrate via nitrification reaction. Nitrate is then transformed to denitrogen thanks to NARG and NOZ genes carried by bacteria. The potential denitrification rates were assessed with measurements of N2O release after 48-hour incubation period with a gas chromatography analyzer. NARG and NOZ gene abundances were evaluated with a real-time PCR technique. The experience plan is to stress the soils at 40 degrees Celsius for 21 days and measure the basal respiration, molecular biomass, phosphatase, beta-glucosidase activities, and potential denitrification rates just after the stress to test their resistance compared to the controls, which were not stressed, and are named TIS, TLO1, etc., due to the French word témoin for control. The soils were hydrated and the functions were remeasured 5 days after stress to evaluate the resilience of functions on the short term. Later, on the same analysis were conducted 30 and 92 days after stress to evaluate the resilience on the medium and long term. Concerning the soil structure, we can distinguish between IS, which is loamy sand, LO1, LO2, which are loamy, and HO, which is sandy loam. This PCA clearly shows the distinction between IS and its control TIS and the rest of the soils along the first axis. It also shows the distinction between HO, LO1, LO2 along the second axis, probably due to its higher total organic carbon content. This also shows that there is no evolution over time to all these parameters between day 0 after stress and day 92 after stress. The molecular biomass of our soils were between 1000 and 8000 nanogram DNA per gram of soil, which is very low compared to the national mean of molecular biomass in French soils, which is 62,000 gram of DNA per gram of soil. The observation of results of basal respiration shows a significant decrease of microbial activity at T0 for LO1 and LO2 soils, implying a weak resistance to the stress. On the contrast, IS and HO have shown good resistance. Concerning phosphatase and beta-glucosidase activities, we can easily see that IS has the lowest activity compared to all other soils. Also, all soils except IS were negatively affected by the stress and showed little resistance. However, HO's phosphatase activity seemed distinctively resilient on the short and medium terms, which is at T5 at T30. As for potential denitrification rates, all soils were negatively affected by stress, as significant differences were found between treatments and the controls for all soils. Nonetheless, all activities recovered quickly after rehydratations and were maintained over time, showing that all soils were resilient. With the exceptions of IS, all soils' NARG gene abundances were significantly lower than controls, which shows that the denitrifying communities were indeed affected by stress. We observed that, contrarily to PDR, activity gene abundances did not recover for most soils, meaning that the suppression of the denitrifying community at a certain degree do not necessarily imply the alteration of denitrification function. No-C gene abundances were also significantly lower than the controls except IS, which confirms the impact of the stress on the denitrifying microbial communities of these soils. However, on the contrary to NARG gene abundances, they recovered 5 days after the stress, which reminds us of the functional redundancy principle, meaning that when NARG carrying communities were suppressed, their niche was possibly occupied by no-Z carrying communities, therefore ensuring the continuity of the denitrification process. We noticed a big difference between T0, T5 on one hand and T30 and T92 on the other hand, which may be due to the installation of the anaerobic conditions in our microcosms due to the prolonged incubation period. This favored the denitrification process, which led to the high denitrification rates. In conclusion, the rehabilitated urban brine-field soils were all very low in molecular biomass content. HO, high organic matter soil, indeed had the highest total organic carbon, total nitrogen and carbon contents, therefore expressed higher potential denitrification and beta-glucosidase activities. High organic matter soil was also the most resistant concerning basal respiration after stress. He was also more resilient concerning phosphatase activity on the medium and short term. The importation of technosols rich in organic matter, even though costly, yields the best results concerning the stability of soil functions after perturbation.